Now it's my turn to give the presentation. I'm Yahdiana Harahap from University of Indonesia. And in the University of Indonesia, we have the Bioavailability and Bioequivalence Laboratory. But in this research, we make collaboration with Clinicindo laboratories in Indonesia. Yes, I'll present about the bioequivalence study of 25 milligram quetiapine film coated tablets in healthy Indonesian subjects, volunteers. So, as you know that quetiapine is antipsychotic agent belonging to the dibenzodiazepine class of drugs, and it is used for treatment of schizophrenia and of mania associated with bipolar disorder. It binds to multiple receptors, most of them with low affinity, and it interacts with dopamine D1 and D2, serotonin 5-HT1A and 5-HT2, adrenergic alpha-1, and histamine H1 receptors. And this is the structure of the quetiapine. Following oral administration, quetiapine absorbed in a gastrointestinal tract, and time to reach peak blood levels for immediate release is 2 hours and 6 hours for extended release. The drug is approximately 83% bound to the serum proteins, and the half-life of quetiapine is 7 hours, and poor bioavailability because about only 9%, and due to extensive first-pass metabolism. In Indonesia, there are many generic products that developed by the manufacturer. In this research, the manufacturer developed a film-coated tablet containing 25 milligram of quetiapine. And due to the regulatory for generic product, it is compulsory to go through the bioequivalence study. So the objective of this research to compare the bioavailability of two 25 milligram quetiapine film-coated tablets in healthy Indonesian subjects. And this is the methodology. Before we conduct the bioequivalence study, the study protocol should submit to Committee of the Medical Research Ethics of the Faculty of Medicine, University of Indonesia, and was approved by the National Agency of Drug and Food Control. The design of study is a single dose, open label, randomized, and two-way crossover in fasting condition. After we obtain the ethical clearance, then we do the subject screening. The subject screening, we do the subjects should pass the physical examination, also the ECG and clinical laboratory testing, such as hemoglobin, hematocrit, white blood count, platelets, white blood count differential, blood urea nitrogen, SGPT, SGUT, alkaline phosphatase, total bilirubin, total protein, fasting glucose, albumin, creatinine, urine analysis. And also, we also check the pregnancy to the female subjects, and all the subjects should negative result of hepatitis B, hepatitis C, and HIV. And the subject will be excluded if they have a history of hepatic, renal, and cardiovascular system, take alcohol or other medication for a long period of time, have hypersensitivity to quetiapine, receive any investigation drug within 4 weeks, and lose more than 450 milliliters of blood within 3 months prior screening. And after we do the screening, we obtain 24 subjects, consist of 19 males and 5 females. And then after we give the detailed explanation about the research to the subject, the 24 healthy subjects will sign the informed consent. This is the demographic data for quetiapine in 24 subjects. The age, the range is 18 to 47, and the weight from 44 to 74, and the height 153.5 to 179, and the body mass index from 18 to 25, the range 18 to 25, and the mean 21.1 plus minus 2.8. And then we do the blood sampling from pre-dose until the 24 hours. It is regarding the half-life of the drugs about 7 hours for 3.5 times. So 24 hours is enough. Before we do the sampling to the subject, all subjects avoided using other drugs for at least 2 weeks prior to the study, and until after its completion. They are also refrained from ingesting alcohol, caffeine, chocolate, tea, or Coke containing beverage at least 48 hours before its dosing and until the collection of the last blood sample. Subjects were confined at clinical unit of Indonesian laboratories one night before study to assure the fasting condition. In this research, the subjects should fasting about 10 hours before the drug administration. On the study day, subjects were given 1 tablet of either product with 240 milliliters of water, and no food was allowed until 4 hours after dose administration. Water intake was allowed 2 hours after the dose, and standard meals were served at 4 hours and 11 hours with the 939 calories and so on, and snacks were served at 9 hours. And the total calories were calculated by the nutritionist. During the blood sampling, the blood pressure, heart rate, body temperature, and adverse events also monitored by the physicians. In this research, there is no serious adverse events, but only mild. We see in this table that there are some events such as somnolence, dizziness, dry mouth, nausea, weakness, hypotension, and polyuria. So the total events about 86. From the 24 subjects, the 23 subjects, these events happened to 23 subjects. And after that, we take the 9 milliliter of blood, and then we do the blood separation. The plasma was frozen at minus 20 degrees Celsius until analysis. And after the subject complete the 2 periods of the sampling collection, and then we analyze it with the validated method in terms of selectivity, accuracy, precision, and then also stability, matrix effect, and carryover. And we analyze it with the LC-MS-MS. This is the mass spec condition. The scan type is multiple reaction monitoring with electrospray ionization, and the detection mass for quetiapine is 384.1, and this is the fragmentation ion 253.1, and this is the internal standard clozapine 327.1, and this is the fragmentation ion of clozapine to 270. And this is the chromatographic condition. We do the river phase with dimension of column 50 times 2 millimeter with particle size 4 micrometers. And this is the gut column. And the column temperature was 40 degrees Celsius. And the mobile phase with gradient elution consists of 0.1% formic acid in acetonitrile and 0.1% formic acid in water. And this is the profile of the gradient elution. The flow rate was 0.6 ml per minute, and the injection volume was 5 microliter. The runtime only 4.50 minutes. Before we analyze it, we do the sample preparation. 500 microliter of human plasma sample was added with 20 microliter of clozapine as internal standard. And then we add 200 microliter of sodium hydroxide and 3 milliliter of ethyl acetate. And then we vortex for 1 minute and centrifuge at 3000 RPM for 10 minutes. After that, the organic phase was removed and evaporated to dryness under vacuum at 60 degrees Celsius for 15 minutes. Then the residue was reconstituted with acetonator and water, and last 5 microliter aliquot was injected into the LC-MS system. After we analyze it, and then we do the pharmacokinetic and statistical analysis, the bioequivalence parameters that we calculate in this research, the AUC0 to T, AUC0 to infinity, and Cmax. Cmax and Tmax we get from the individual drug plasma concentration time data and were used as measure of rate of absorption. AUC0 to T was calculated using the trapezoidal rule, and K elimination we get from least squares regression from the data of the last 3 until 8 points of each plasma concentration data curve. And the half-life ln 2 divided to K elimination. And for AUC0 to T, AUC0 to infinity, and Cmax, a multiplicative model was assumed, and analysis of variance was applied using the respective learned transform data. And the bioequivalence, the 90% confidence interval of the geometric mean test to reference for AUC0 to T, AUC0 to infinity, and Cmax were calculated assuming a multiplicative model. The accepted bioequivalence range for this parameter was 80 until 125%, and all statistical analysis were performed using EquivTest version 2 software. This is the mean plasma concentration profiles of the two formulations in 24 subjects. We can see this is the test product and this is the reference products. If we see from this curve, we can assume that there is no significant difference between the two formulations. This is the reference product from AstraZeneca. And this is the parameters for Cmax. We see in the test formulation, the concentration 71.53, and this is 75.03. And for AUC0 to T, for test formulation, 266.94, while reference formulation, 298.27. And for AUC0 to infinity, 272.09, and reference formulation, 304.46. And the half-life for the test formulation is 3.69 hours, and reference formulation, 3.93 hours. And Tmax, same, that was 0.75 hours. And this is still the evaluation. This is the test to reference point estimate for Cmax, about 100.09. The confidence limits range is 93.31 to 107.36, and the intra-subject CV is 27.39. And for AUC0 to T, the test to reference point estimate is 99.89. The confidence limits range 94.89 to 105.15. And the intra-subject CV 21.66. And for AUC0 to infinity, the test to reference point estimate is 99.90, and the limits range 94.85 to 105.23, and the intra-subject 21.68. So from the research, 90% confidence intervals of AUC0 to T, AUC0 to infinity, and Cmax ratios of quetiapine were included in the range of bioequivalence 80 to 125%. The mean ratio of AUC0 to T to AUC0 to infinity for all individuals and for both products was around 1%. It is indicate an adequate sampling time since the extrapolated portion of the total AUC is less than 20%. The intra-subject variability of quetiapine in AUC0 to T was 21.66%. It's also indicated that the sample size of 24 subjects was sufficient in order to conclude bioequivalent with the power of 80% at the 5% nominal level that we get from the literature Diletti et al. 1991. The Tmax was not statistically different between the two formulations, and the median Tmax for both formulations about 0.75 hours. From the literature, from the previous literature, we see that the Tmax is 2 hours. The half-life in the study, 2.45 to 6.53 hours for the test product, and for the reference product, 2.57 to 7.89 hours, while the result reported in the some literatures about 7 hours. This is the chromatogram of blank plasma, and this is the chromatogram of a lower limit of quantification of quetiapine in plasma, and this is the chromatogram of low quality control of quetiapine, and this is the chromatogram of medium quality control of quetiapine in plasma, and this is the chromatogram of high quality control of quetiapine in plasma, and this is the chromatogram of quetiapine in subject sample. So the conclusion of the studies, the two quetiapine formulations were equivalent with respect to the rate and extent of absorption, and it can be assumed to be therapeutically equivalent and exchangeable in clinical practice. And thank you very much. And in this moment, I would like to thank the University of Indonesia that have given fund to join this this conference, and also to the Clinics Indo Laboratories who has funding this research. Thank you very much for your attention. Any questions?
This study is done on the patient or in the health volunteer?
Yeah. Not in patient, but in health subjects.
Okay. Well, because I think a lot of like a psychologist drug, the patient may have different
Yeah.
on the healthy volunteer. So here you say that your Tmax about 0.75. Is that possible because it's different from the literature of 2 hours because
Maybe about the genetically different, because the literature we get from another nation, not Indonesian, but in the two formulation is similar. The Tmax is similar. Yeah. Yes.
Is blood sampling using bedside or bedside sampling has been done in this study?
Yeah.
Bedside sampling, blood sampling.
Blood.
Bedside. The moment you administer the investigation product, the blood sample, PK samples.
Yeah.
You collected on the bedside or at the, phlebotomy area?
Oh, the site. Yeah, the Indonesian laboratories.
No, sampling.
Yeah. Yeah,
You
we
did
do.
at the bedside. The beds, volunteers are housed in beds, right?
No, the sampling we do to the subject in the laboratories. Yeah. In the laboratories, there's the site for the blood sampling, and also for the quarantine, and also for the preparation room, and also for the analysis the sample. So in one site.
Being an antipsychotic drug, adverse events are more.
Yeah.
So the frequent, movement of these subjects will inconvenience.
In this study, we made also collaboration with the hospital near to the laboratory, and also there is a physician in the site.
Yeah, how many times the sampling time was?
About 15 times from, let's see, sampling time. This. Yeah, about 15 samples, 15 times.
This is according to which guidelines?
Yeah. For the guideline between 12 until 18 in Indonesia, we have the guideline. The samples for the subject is between 12 until 18 times. Yeah. And we collect the blood until 24 hours because the half-life is about 2 hours. Yeah. So we can do the blood sampling 5 to 2. Actually, we can only until maybe 12 hours, but we want to do until 24 hours. So the AUC 0 to T and AUC 0 to infinity, not different larger. Yeah.
Do you have any experience with steady-state bioequivalence studies with quetiapine in healthy volunteers?
No, I don't have the experience for.
Did you perform?
Dose,
Did you perform any study with a higher dose than 25 milligrams? Higher dose, maybe 50 to 100?
No,
No?
not yet. If we want to do this research to the patient, maybe it's better we do in multiple dose in schizophrenia patients. But in Indonesia, we haven't done like that. So this is to the healthy subjects and only single dose. Any questions? If no more questions, so maybe you can give me applause.